The overall goal of this procedure is to generate and isolate AAVS1 safe harbor targeted inducible pluripotent stem cell, or iPSC, clones expressing GFP. This is accomplished by first delivering AAVS1 TALENs and AAVS1 CAG EGFP donor plasmids efficiently into iPSCs by nucleofection. In the second step, the AAVS1 targeted cells are selected by puramycin on mouse embryonic feeder cells. Next, the individual GFP-expressing AAVS1 targeted clones are picked into a matrigel-coated 96-well plate. In the final step, the AAVS1 GFP iPSC clones are expanded with E8 medium and standard iPSC culture techniques. Ultimately, immunofluorescence microscopy and flow cytometry are used to show the uniform and robust expression of GFP in AAVS1 targeted human iPSC clones.
Visual demonstration of this method is critical, as the colony picking steps involve microdissection of targeted clones using special colony picking tools under a microscope, which is difficult to learn during side-by-side instruction.
Begin by washing the iPSC cultures one time each with PBS. Then add 1 milliliter of 37 degrees Celsius gentle cell dissociation reagent to each well and incubate the cells at 37 degrees Celsius for approximately 5 minutes. When greater than 50% of the cells have dissociated from the culture